All right, part 3 of chapter 12, still on adaptive immunity. Here we're looking specifically at cytotoxic T cells. All right, what is their role to eliminate antigens? And if we're talking about a host cell, if it is infected with a virus or if it is cancerous, it can signal the cytotoxic T cells, and because those cells express MHC class 1, the cytotoxic cell can come in and attach to that cancerous cell or virus-infected cell and secrete different materials on it to actually cause it to die. And on the right side here we see that the T cell receptor, when it binds to an MHC class 1 with the antigen, it's going to release perforins, and that creates a pore. Right, the perforins are a collection of proteins that will bind together to create a pore. And then it will also, the T cell, cytotoxic T cell, release granzymes. And these are material that will move through that pore and break down host proteins. And in that process, it will induce apoptosis, and that cell will die because of the actions of the cytotoxic T cell. All right, and this is a picture of what's happening. Here you have a cancer cell or some sort of infected cell. It secretes signals, it will release interferons. Interferons will activate and recruit the cytotoxic T cell, and it will also express antigens on the surface in conjunction with class 1 MHC. And the cytotoxic T cell will come in and it will release perforins. All right, and these perforins create this pore, all these orange pieces, and then the granzymes will come in and enter the hole, the pore, created by the perforins and cause that cell to undergo apoptosis. All right, and then macrophages, which are over here hanging out in the periphery, will come in and clean up that garbage. All right, helper T cells will play a role in antigen elimination by being able to help out the other arms of the immune response. All right, so supports the action of the cells that it will actually do the work, and those are going to be the B cells, the cytotoxic T cells. And remember that T helper 1 cells favor the cytotoxic T cells, whereas T helper 2 promotes the humoral response. All right, so then what is the B cell response, that humoral response? Right, it goes through the same major steps as T cells in terms of being activated. All right, and here we're looking at specific interaction of this blue circular antigen with the different clones of antigen or of B cells that are out there. And it doesn't fit here, it doesn't fit here, but lo and behold, this specific interaction is going to occur. And then this one B cell gets activated and it proliferates to increase in number, and then it differentiates into plasma cells or memory cells. And the function of the memory cells is to be long-lived, and those will be available to activate quickly if this same blue antigen, so to speak, comes back into the body. This process here is actually going to take that 10 to 14 days. All right, but once those memory cells are made, then this response the second time around could actually only be like a day or two. All right, and the function of antibodies, all right, we'll look at that. The function of plasma cells is to secrete high numbers of antibodies. And then the functions of antibodies, this really is the effector function of B cells, even though it's technically at this point a plasma cell. Antibodies can neutralize antigens, all right, so it can bind the antigen here, and then this antigen can't bind to the host cell. If it can't bind to the host cell, then it doesn't do anything. All right, so it's neutralized. All right, then another step is to activate complement. All right, and in that process, the activation of complement can allow for the MAC, all right, creating holes in the pathogen. All of these purple pieces are complement proteins that will allow water to go in and cause lysis of that cell. You can kind of see that here. And it going through this process, it ruptures and it dies. Opsonization, the complement binds to the pathogen, and that helps the efficiency of phagocytosis. And then complement also leads to inflammation, and we looked at that, all of these different processes in chapter 11, all right, the innate immune response. All right, complement is part of the second line of defense, even though it's activated by antibodies in the third line of defense. All right, and then increased efficiency with phagocytosis. All right, if an antibody binds to the antigen, all right, this antigen is stuck to the cell, all right, then it can cause agglutination, and agglutination is just making clumps. And then opsonization, same process kind of as over here, all right, right here, and the phagocytes can increase its phagocytosis process. All right, so what does an antibody look like? What is the structure? All right, the structure is this Y-shaped protein here. All right, there are 2 heavy chain pieces. That's this large piece. It's heavy, it's bigger. And then there are 2 light chains. So it's 4 pieces of protein stuck together. Those are stuck with disulfide bridges. All right, and in this area, part of the light chain, part of the heavy chain create the antigen binding site. And so this is, the entire antigen, and on this antigen, there's this epitope, the triangle, epitope, square, and circle. This antibody is going to bind specifically to this epitope, and that's going to be based on genetics, and this is actually called the variable region of the antibody. And even though both of these antigen binding sites on this one antibody are the same, you can have over 1 million different types of antibodies that are in your body at any given time available to bind different epitopes. All right, now there's different forms, isotypes. All right, there's a typo there. Not isotope, isotype. 5 major antibody isotypes. There is IgG, IgA, IgM, IgE, and IgD. All right, what we're going to focus on is these notes section. All right, IgG is the most abundant antibody in the serum, followed by IgA. IgA is found in mucous membranes also, and then IgM is the third most abundant. You don't see IgE a lot, IgD even less. There's actually not a whole lot known about IgD. IgE is important to fight against parasites, but it also mediates allergic reactions. IgM is made very early in infection. It can be a pentamer, which has 5 pieces, 5 of the same IgM antibodies stuck together. IgA is the main antibody that is secreted in breast milk, and that is one reason why it's good to breastfeed an infant, because their immune system is weak when they are first born. Their immune system needs time to mature, and if the mom breastfeeds, then she is giving her antibodies to the infant to help protect that infant until the infant's immune response can do its own thing. And IgA can be a monomer or a dimer. All right, there's two of them stuck together in the middle. IgG crosses the placenta. That's the only one that can cross the placenta, which means in utero, the infant is getting some of mom's IgG, and that also helps protect the infant in those first few weeks, first few months of life when the newborn's immune response or immune system is trying to figure things out. All right, here's another picture of antibody functions where there is neutralization here. All right, the antibodies bind to viruses and neutralize them. If the virus cannot bind to the host receptor, it can't get into the host cell, and therefore you're protected. So this is what is happening when you get the influenza vaccine. When you get the vaccine, your body produces antibodies so that then if you get exposed to the virus, those antibodies can neutralize the virus before it gets into your cells and actually makes you sick. All right, there's antitoxins. It's going to neutralize a toxin. All right, very similar idea with neutralization, except it's in this case because it's neutralizing a toxin, it's called an antitoxin. All right, here is causing lysis of bacteria cells. That's because it's activating complement. And then this is the agglutination, making those clumps. When you have those antibodies, remember the antibody has two antigen binding sites, so it can hook one bacteria cell up to another. This antibody is keeping two close together. All right, this allows for basically sequestering of the bacteria cells. They can't get away. Makes it a little harder for the bacteria to actually replicate when it's clumped up like this. And then if it's all clumped together, it keeps everything in one spot so that when phagocytes come in, they can just do their work right here and get it done quickly. And this is the process of opsonization, which makes phagocytosis more efficient. Alright, so this is just a fun review slide. You can look at this on your own. Alright, and then in class on Thursday, we will look at immunological memory. Alright, in terms of your immune system's response the first time it sees an antigen and then the second time, and this is what's going on. Alright, and we'll also look at humoral immunity, which is active and passive, and that's it for part 3.